Okay, welcome everyone. So this is our first machine learning and computer vision seminar here at LCLS. Essentially, the idea of this is to showcase exciting work that has the potential or has already demonstrated usefulness for tackling the data scaling, data reduction problem that we are already facing and about to face even more at LCLS. And so I think it's very fitting that Kevin does the inauguration of this seminar series. I don't know how we are going to organize this, but this is the first one and maybe the last one because it solves all the problems. But we hope to hear about a lot of other developments in the future. But yeah, like I said, very fitting to have Kevin present today about his work. Maybe a few words about Kevin. So Kevin, you did your PhD at Stanford in biophysics after having been trained as a chemist and a biochemist. And after that, you moved to Docker Extra Lab to work on developing method and experiment for multi-temperature and time-resolved crystallography, which is very important to us here at LCLS. And so we are very pleased to have you now among our ranks. And please take the floor, and very excited to hear your presentation. Thank you.
In certain work environments, harmful dusts, mists, fumes, gas.
Can everybody see my screen?
Yes.
That's a yes.
Okay, good. All right. Yeah. Thank you, Fred, for the introduction. Yeah, I'm Kevin. I'm a relatively new hire at LCLS in the data systems group. And what I wanted to talk about today was a project that I started during my postdoc with Docker Extra. And really, it's about taking work that I did at sort of the middle of my postdoc, which was only applicable to relatively small data sets, and making it scale to the sort of data sets that LCLS is producing. And so I don't really need to tell this group, but we have an enormous problem with modern light sources, that they're producing sort of enormous data rates. I really like this video from Max Vidorn from UXL running in the megahertz mode. You can see the beam cutting into the liquid jet of the sample. It's super fast. So this is a big challenge to keep up with in terms of data analysis. And there's really two ways that I'm aware of to deal with these kinds of large data rates. One is by getting a really big computer, right, and writing parallel algorithms, which can be a very successful strategy. And we're very fortunate to have collaborations with many national labs that have such computers, and we have our own computing resources at LCLS as well. But what I wanted to talk about today is less that sort of brand of scaling, and more in the line of looking at our algorithms and seeing if we can make them more efficient, make them just natively handle larger data sets without resorting to really large supercomputing resources. So what I'm going to talk about today is in the domain of X-ray crystallography, in particular serial crystallography. And most of you should be familiar with the sorts of, how the data look in crystallography. We collect these images. They have spots in them called Bragg peaks. And one thing that you do very early in data analysis is you go and you predict where all the spots should have landed on the detector. You visit them and you estimate the photon flux scattered to each of them. So that's a task that is usually referred to as integration. That gives you a list of tabular data like this, where you have intensities, which are your flux estimates, usually error estimates to go with them, and various metadata about each observation. If we lived in an ideal world, we could take this tabular list of data and just average all of the redundant observations, and then take the square root to convert them to structure factor amplitudes. That's the thing that we want in the end of the day. And then we can go about our business of phasing those structure factor amplitudes and do the model building where we fit atomic coordinates to the electron density that we got from phasing. Most of you should appreciate that this doesn't sort of work out of the box. You can't just do this, and the reason is by the very nature of the design of the experiment and the quality of the sample, diffraction images are just sort of contaminated with a huge array of systematic errors. I like to visualize this by taking, in this case, conventional rotation method diffraction data and plotting the histogram of intensities as a function of various metadata. So in this case, you can see that the trend in intensity varies as a function of where you are in the detector from this like X and Y coordinate on the detector. And indeed, it varies throughout the data set in this kind of jagged, wildly nonlinear way. So as a community, we've developed a class of algorithms that we refer to as scaling algorithms. And they take this kind of jagged, corrupted view of the data and smooth it out to something like this, okay? So that slots into the data reduction pipeline right after integration. You can correct the systematic errors using a scaling algorithm. Then you can merge the redundant observation. Those can be fed back into the scaling algorithm as part of the objective. You can iterate this until convergence. Only then can you convert the merged intensities to structure factor amplitudes. We do that with a Bayesian protocol, not simply by taking the square root. The reason we have to do that is because negative intensities happen statistically just by chance from time to time. So you can't take the square root of a negative number. So we had to invent this Bayesian protocol. Right, then you can do phasing and go about building your model. That's sort of data reduction pipeline in a nutshell. I'm going to be talking about scaling mostly today. And I wanted to highlight, because we're at LCLS, that this is a particularly acute challenge in the free electron laser space. And that's because free electron lasers have a lot more systematic errors than conventional diffraction data. One simple reason for that is because the beam intensity fluctuates a lot more than a synchrotron, as you can see in the single crystal data that we collected at XDP. This was a crystal grown by Ian White in the Brunner lab. And you can see the beam intensity just bobbing all over the place, even though it's the same sample. It has nothing to do with the sample, or there's no liquid jet here or anything. That's just the fluctuation in the input photons. But that's not really that hard to correct for. That's not the big issue. Really, the more insidious kind of systematic error you run into in free electron laser data are partialities. So in a conventional rotation method experiment, you get to enjoy what we call full intensities. And you can do that because you have a long enough exposure time that you can rotate the sample during data acquisition, and you can integrate the full diffracting volume of each fragment. And you can't really do that if your exposure time is in the femtosecond regime, right? You can't spin a sample fast enough. So you end up with some ill-defined slice through the diffracting volume, and you need to try to extrapolate back to what the full intensity would have been. That's what we refer to as the partiality problem. So when you're doing scaling, you have a lot of choices of different algorithms, different packages, and the one you choose is going to be dependent on what sort of data you have, whether it be conventional serial, be it at a synchrotron or a free electron laser or polychromatic data. The major research product of my postdoc was this algorithm, which takes all of these separate scaling models and it generalizes them into a single model. So that's this graphically represented as a probabilistic graphical model. And we implemented that in a package that we called Careless. And to be transparent about it, what Careless does is it takes this unmerged, unscaled, meaning containing the systematic errors still, integrated data, the intensities, the error estimates, and also the metadata. And it converts that into merged structure factor amplitudes. So revisiting the kind of data reduction pipeline here, that it's really doing all of the center strike here in one single optimization algorithm. And the way it works is really, I think, pretty intuitive. It has to do with the fact that if you're looking around in this high dimensional space for the correct set of structure factors, you can show from statistics that you'll know that you've obtained them if you can satisfy these two criteria. Okay, first, they need to be consistent with the data you observed. Obviously, why else, why did you do the experiment if you're not going to use the data? So it should be that the structure factors you estimate are consistent with the observed data. And then they should also be consistent with our expectations from first principles. So in Bayesian language, a prior distribution. And we're very fortunate in X-ray crystallography to have this very innocuous choice of prior distribution, which is still pretty informative. And that comes in the form of the random atom model, which is the distribution of intensities that you would expect if you just had uniformly distributed the atoms within the unit cell of the crystal. It turns out you can work out distributions for that case, and they've been used very heavily throughout crystallography and to a lesser extent cryo-EM too. So in Careless, that's what we use. We plug that in as the prior distribution. And we know from statistics that we can recover estimates of structure factors like this that satisfy these two criteria by maximizing the subjective function. It's called the evidence lower bound. It has two terms, and they just work like this. Right? The first term ensures that the structure factors fit the data, and the second term ensures that they don't wander too far away from the prior distribution, from the random atom model. And this would work kind of out of the box without any modifications if we didn't have to worry about this systematic error issue. That was a major challenge in implementing this graphical model. So we had to figure out, okay, how do the systematic errors come into this? And it turns out that they really only impact this first term, the expected log likelihood term in the elbow. And the way we chose to handle it was pretty generic. It was not by writing down a physical model that said, okay, we know this about this type of experiment. So, we're going to compute based on a physical model, like how the systematic error should depend on, spatially on where you are on the detector or whatever. It wasn't like that. We didn't say we're going to fit B factors to every image, stuff like that. Instead, it just starts from the assertion that given a sufficiently rich set of metadata about each experimental observation, there should be a function by which you can compute an estimate of the systematic error. We don't necessarily know what it is up front, but we think it should exist. So what I did was to just say, okay, we're going to have a neural network that takes in metadata about each observation. It ingests a vector of metadata for each reflection observation, and it's going to output an estimate of the systematic error for that observation. And then we're going to use gradient-based optimization to maximize this elbow objective function with respect to the structure factors, right? That's the thing that we care about, getting out the back end. and the weights of this network. So we're going to jointly, for each data set bespoke, learn a model that takes in whatever metadata you give it, which is hopefully encapsulating all the sources of systematic error in some sense, and it's going to output an estimate of the systematic error. And this, to be fully clear, this is going to be retrained for each data set. And I showed in the original publication that this could be applied to serial femtosecond crystallography data, so that it could address this partiality problem that we discussed before. I showed that using this zinc metalloprotease data set. So this enzyme thermolysin has a zinc atom, a zinc ion in the active site. And that's a source of anomalous signal that we can use to validate the quality of our scaling results. These were data that are publicly available. They're deposited in the CXIDB. They're processed by Aaron here. Yep. And for this study, I had just taken a small subset, the first run, which was about 3,000 images. And the way that I was able to convince the model to solve the partiality problem was to take the refined geometry for each image and compute an estimate based on that for how far off the diffracting condition, the ideal diffracting condition each observation was. So we calculated the length of this vector here, which we called the Ewald offset, that's EO. And you simply supply that as an extra column in the metadata. And I showed that if you do that, you can get results for this anomalous peak height, which are comparable to sort of more conventional approaches. And then critically, if you ablate that function by removing that metadata column, you don't do so well. You do really quite poorly. So the model was really able to take that metadata entry and figure out how to correct for partiality based on that, and without that it couldn't. So this worked really well, we thought, but there was a real issue with it. I had to kind of cheat to get it to work. So cards on the table, this was the hack that I used. I took this nice, simple multi-layer perceptron scaling model that we use by default in Careless, and I added this feature that we called image layers. So these were just like normal layers in a deep neural network, in a regular perceptron. But they have separate weights for each of the images in the data set. And this allows the model to learn some sorts of corrections that are bespoke for each image in the data set. So we added some local parameters for each image. And this wasn't a problem for that small 3,000 image data set, but you can appreciate that if you're going to have the number of weights scale with the number of images, that's not a good idea if you want to be merging hundreds of thousands or maybe millions of images. And this was a really material limitation for us. So this is the largest data set that we ever analyzed with Careless. This is a sensory rhodopsin data set that was provided to us by Richard Noitz's group. They had about 100,000 images or so, half of them in the dark state and half of them in the light state. And we were able to process these data, but in order to do so, we had to allocate a big memory node on the Harvard cluster. So one with half a terabyte memory, and it took about a week to train the model. So this is not something you could do during a beam time. It's not something you would even want to do routinely, really. So to really make this useful, especially in the serial crystallography setting, What needed to be solved was how can you replicate this functionality of the image layers, but without having any local parameters. And what I settled upon was this kind of encoder model, where you have an image encoder that takes in the metadata, and it takes in also the intensities. So it's kind of got access to the answers, of not just the metadata, and it creates a vector that represents each image in a particular batch of data. And then when you go about doing scaling, you get the metadata as you would in the normal model, but you also get access to this kind of compressed representation that's supposed to have all of the relevant information about each image. So in a little bit more detail, and don't worry if this is overwhelming. The architecture of the model is, you know, you don't need to understand in great detail if you're not a deep learning person. It's fine. The important thing to note is, like, there's this kind of two branches to the scaling model. One gets access to the intensity and the metadata, and it outputs a vector that represents the content of the image. And then the one which actually the branch that estimates the systematic errors gets access to the metadata and this compressed representation of the image. I've applied this to a few test cases so far. These again are all publicly available data sets from the CXIDB. One of them is again that zinc metalloprotease data set from Aaron. The other two are from Sakla. They kind of have a bit of diversity in the data set sizes. One is notably smaller than the others. They're all relatively high resolution. This is the only one processed in dials, the other two in Christfell. But importantly, they all have anomalous scattering centers that we can use to validate the fidelity of the scaling. And so that's what I've done so far. Is I've run the code on each of these examples, and I've looked to see how does the anomalous peak height that we get compare to the published values. You know, I think it's doing competitively so far for these three examples at least. I'm sure that we'll find some for which it doesn't perform so well, and we'll probably learn things about the architecture by doing that. But this is the kind of gallery of examples I have for the time being. And I went back and I compared the performance to the previous model, the one with the image layers. And this is, again, it's just a small, about 3,000 image or so subset of this larger CXIDB81 thermolysin data set. And it performs pretty close. The encoder style model has pretty similar performance to the image layer style model, which means that, you know, this new architecture scales to very large data sets without too much difficulty, but it can also be useful for looking at smaller data sets. And one interesting challenge that I had to address when I was developing this gallery of examples was this CXIDB62 entry, which is, I think it's a sugar binding protein from a mushroom. Anyway, you never know what people will be interested in. It's in this space group P65, which is one of these unfortunate space groups that has marahedral twinning, which means that even if that particular crystal form isn't twinned, you do have this indexing ambiguity problem. That means that in serial crystallography, a given image, the crystal could have been in either one of two orientations, and you'd see the same pattern of spots on the detector. So without looking at the content of the reflections, you can't know what orientation it was in. So the indexing algorithm is going to randomly pick one. So you have to deal with that downstream. And I solved this problem by noting in this framework that in the objective function, it's only this first term that depends on the orientation. And so what I did was I just said, okay, like let's pretend the crystal was this way and let's pretend it was the other way, and calculate the expected log likelihood of both, and then just pick the one that's bigger. That's it. That's the whole trick. So the gradients only flow from the better estimate. And this worked really well. This is just the same plot I showed before, except it has this new column here, which is if you take this index disambiguation, this take the maximum likelihood of the two possibilities nuance to the algorithm out, and you ablate that, you can't really get a good anomalous peak height out anymore. So that algorithm does seem to work, and it I mean it seems to work well, and if you ablate it, your performance goes way down. And just to be completely transparent, this is not a new idea. I totally ripped this off from this is the same thing as the symmetric loss function that's used in cryo AI. So just to sort of give an idea of the impact of this, I wanted to close by revisiting the largest data set that we processed with Careless. So again, this is that sensory rhodopsin data set, about 100,000 images. It took us a week to merge before. And with the new code, you know, the runtime here, you see the time in minutes versus the number of optimization steps. We can do about 30,000 steps in under 40 minutes. And I'll show you on the next slide that we're quite converged at that point. And one of the exciting things is that not only is this like 100x improvement or so in runtime, we're only using about 4 gigs of accelerator memory. So you can run this merging code on, you know, really consumer grade hardware. It doesn't need a really fancy GPU like an A100 or an H100. And the performance won't suffer very much in terms of runtime. And yeah, just to show how training progresses over those 30,000 steps, you can look at the progress on the loss function on the right. So right, we're trying to maximize this elbow, and it's plateaued by, you know, around 10,000 steps. You're only making incremental progress after that. And on the left, you can see how the structure factors evolve in this like amplitude versus uncertainty space throughout training. You can see they're pretty well stabilized by the end of this 30,000 steps. And then if we look at the output, we get a reasonable electron density map. This is the chromophore, where the photon changes the conformation of this chromophore, and that's how the protein senses light. We get nice difference electron density peaks in this region. The 2 of the 4 largest peaks are in this region, and yeah, they're over 5 sigma. So this is a pretty comparable result to what we were seeing with the week-long training run in Careless. Yeah. So yeah, this is kind of where I'm at right now. This project is exiting the research stage right now. I need to do a bunch of hyperparameter tuning and trying to figure out how to run the model best on average for most data sets. Need to work on developing some more user-friendly data validation tools. But it is out there. It's open source. It's written in TensorFlow. It has a command line interface with, you know, help streams that you can look at if you want. And the goal is to make this available to users at LCLS, you know, soon-ish. I think Fred and I have been discussing, you know, before before the new year, ideally. And that's all I had on this project. Just close by, thanking my sort of old home in Duca's group. Duca has been a fantastic mentor, and I look forward to continuing collaborating with him throughout, in my new role at Slack. And I would say that the three members of Duca's group who contributed most to this work are Jack, who is a graduate student who's now at D. Shaw Research, Rick, who's still in the lab working on Lowy data analysis tools, and Milan, who's like the other big machine learning enthusiast in Duca's group, who's going to graduate in the fall and join the Flatiron Institute here in Manhattan. And yeah, my new home at Slack, Fred and Yana, thank you very much. I guess I'll close there and do questions if anybody has any.
Thanks, Kevin.
Yeah.
It doesn't get old. Like, I've seen it twice now and I still love it. Right. So yeah. Please, guys, don't hesitate to go for questions. I think there is a lot.
I have a quick question. So you said that the two main challenges are the partial intensity, but also the varying shot-to-shot intensity. Have you considered using the beam diagnostics, such as the IPMs or data like this to address especially the second problem?
Yeah, we've thought about this. We thought about this quite a lot at the beginning of my postdoc when we were trying to tackle these problems from a more physical perspective, but we haven't really circled back to them. We haven't been in a position until recently where we could put very high dimensional metadata in, but we can now. And so I think it'd be really interesting to put entire spectra in.
Can I share a thought about
Thanks.
the specific idea with the energy and the pulse? It's going to be totally correlated with the illuminated volume, and that is totally unknowable. So there might not actually be any real point in normalizing to the pulse energy.
And that's due because of the pointing jitter or your sample volume that varies shot to shot?
Yeah, the crystal size, and I mean,
Okay.
maybe besides that, you hit the corner of a crystal or something.
Makes sense. Yeah.
There's always a scale factor that you don't know.
Mm-hmm.
Yeah, I think I'd be more interested in incorporating the spectral information for the safety
Right.
spectrum.
Mm-hmm. That makes sense. Thank you.
But actually, for things like the IPM detectors, putting the position information in could be useful.
Mm-hmm.
When we were doing those single crystal experiments at XPP, we did show that we could correlate the IPM values to the intensity we observed on the detector. So I think there is some info there.
I think Sandra, you have a question?
Yeah. So first of all, Kevin, thank you so much for the great talk. I really enjoyed it. For me, it was the first time. I would love to hear it a second time. So please let me know. I was wondering, maybe I missed it. Maybe it's obvious, but how does the runtime scale with the number of images?
Oh, so it shouldn't scale with the number of images. So I don't have the number off the top of my head, but I can calculate it. But that runtime reflects many passes over the data. So the data have been recycled through the model in batches many times.
Okay.
So the runtime scales more with the batch size, the resolution, and whether or not you're doing this index disambiguation. That adds, it basically doubles the gradient step time because you have to calculate the loss function two ways.
Yeah, that makes sense. Other than that, I was wondering, so in the past, I often used the Unity model in Crystal for processing data sets. Do you have an estimation? Like I can imagine if you use partiality, have a good partiality estimation that you need many like less images. Do you have an estimation of like how much smaller a data set can be to get accurate structure factors? Or
I mean, I could say something off the cuff, but I don't have any like really justifiable, well thought out answer. But I'd be happy to collaborate on answering that question. I think it's really important.
okay, yeah, I maybe actually have a data set of like 1 million images with indexing ambiguity and anomalous signal. So if you have some time, I would actually love to have that data set processed as well.
I think Aaron's on this call. He did a bunch of work analyzing how anomalous signal goes up in Dial's processing as a function of how many images you include. I'm not aware of any really thorough exploration of that using Crystal.
Okay.
But that doesn't mean it doesn't exist. It just means I should read more papers probably.
Okay, thank you.
Yeah. Vidya, did you have a question too?
Yeah. So first of all, great talk. I've heard it before, but I agree that it was good the second time too. So okay, two questions, I guess kind of maybe like more minor points. Could you explain that there's that graph that you had with the uncertainty and the amplitudes? I wasn't sure what we were supposed to get from that animation. So maybe this is obvious to everyone, but could you explain like what are we supposed to get? Like, yeah, what are we supposed to get from that animation where it kind of goes to that squiggly shape from the blob?
Yeah. Does it help if I pull it up again? Maybe not. Yeah, this was simply, it is a space that you can plot the posterior in. And I just wanted to show that as the objective function levels out, they stop moving around. This could have equally been some kind of other choice of reduced dimensionality representation.
Okay. So there's nothing that we have to get from like, because it seems like the uncertainty is actually like going up as we.
Yeah, so that's pretty much down to how you initialize the posteriors, which direction it moves in this space. I found that for these kinds of variational models, I prefer to instantiate the posteriors so that they're quite narrow, because I find it takes less time for them to broaden out than it does for them to sort of shrink down, and it takes fewer MC samples.
Okay. That's a very good intuition to know.
They're just like hard won kind of insights you get from messing around with these things for way too long.
Okay, that could actually be useful for other projects.
Yeah.
And then okay, so yeah. And I had one more question, which was so yeah, thanks for that explanation. That makes sense. And then the other question was, so I know you started this project kind of a while ago, which makes sense for TensorFlow, but I was just wondering if you thought about switching over. I feel like now PyTorch is kind of the main tool. Like, have you thought about switching over, and like what's kind of your logic behind choosing the TensorFlow framework, or like keeping with the TensorFlow framework, I guess?
So I have the beginnings of a PyTorch implementation, and I do intend to sort of. It's unfortunate, but I will end up rewriting this whole thing in PyTorch. The reason that I've been so sluggish in moving is because there's a few reasons. The way that Paris handles losses, where any layer can sort of like add a loss into the overall objective function locally without having to pass it back to like, the top layer in the model. So that's one thing, but that's a feature that I'm going to work on re-implementing in PyTorch. The other thing is there were one or two distributions that were available in TensorFlow probability, which still aren't available in PyTorch, which I was relying on. So we can talk about this offline in greater detail, but we've got a project on RS Station, which is RS distributions, where we're working on re-implementing some of those. So we have a reparameterized implementation of the folded normal and a reparameterized implementation of the Rice distribution, which I like. You can't really do good crystallography statistics without those distributions. So I was kind of waiting on getting the bandwidth to figure out how to do that, so myself and Luis, who I think is on the call, and Minhwan, we re-implemented those in Torch in the spring at some point. Yeah.
Okay, got it. All right. Okay, that makes sense. Yeah, thank you.
Well, if there is no other question, I might ask one. I mean, I was trying to see what I didn't ask you yet. And like hypothetical, like it might be of interest for some people on the call. Is like you said that you train it for a given data set. So once it's trained, it's not going to be applicable to a new data set. You have to retrain from scratch. And that's a very like current thing for unsupervised models. But like, what would be the way forward to make it more generalizable? Or maybe like, can we talk a little bit about using it in inference mode after having been pre-trained on some subset of data? Those type of questions.
Yeah. So I would say that there were two things missing. Now there's only one. So the first thing that was missing was having a parameterization for the scale model that had only the global parameters. So there would be no way to conduct transfer learning, let's say, if you have per image parameters and then you try to apply it to a new data set that doesn't have that image, it's not possible. So with the global model, I think that we can probably do transfer learning at the very least. Then to get to a point where it can run in inference mode, what we would need is to figure out how to train it contingent on some estimate of merge structure factor amplitudes for each data set. So it would mean coming up with some new training infrastructure. And then I think you probably apply it quite differently. You probably wouldn't do variational inference to estimate this, the structure factors. You'd probably use it more like a conventional scaling model and correct the intensities and then average them in a maximum likelihood way.
Right.
And that would be, you know, instantaneous to apply. But you have to have a source of merge structure factor amplitudes that you trust.
Right.
That's
maybe, yeah. So maybe like after a few runs, you converge, and then you're like, okay, now we can do maybe not for the full analysis, but maybe for some live feedback, we could just have a quick update of the maybe that would help with the signal to noise or something like that, right? Like running in inference mode.
Yeah, I mean, it would be a cheap way to run a scaling model.
Right.
But if you want, like what you'd like to do in an ideal world is train it across a whole bunch of data sets and at input high confidence estimates of the merge structure factor amplitudes for each of them, and just train the scale model.
Right. Yeah, okay. So maybe things we could look into. All right. No burning question. It's now or never, but no, I mean, we can always follow up. Well, yeah, sorry.
The most important thing that I learned from Duca during my postdoc was that nobody ever complained of a meeting being too short.
Right. Yeah, and that's convenient because yeah, we have to go in 2 minutes, so that's perfect. Well, in that case, well, first I want to thank Kevin again for this great talk and I think exciting tool for LCLS and other facilities. But yeah, also for having the inspiration. I think there are I've been lurking around in the group here, and I think it's a bit wider than usual. So I'm very happy to see people from outside of crystallography paying attention to this, and hopefully we can cross-seed ideas over the next few months and years. And yeah, thanks, Kevin. Thanks everyone for joining. I'll stop the recording now, and I wish you all a great rest of the day.